My name is Maria Maccecchini. I'm the moderator today, and I'm very, very happy that we have both our speakers and that this February Joseph Priestley Society program features two people that are at the forefront of alternative methods of drug delivery, Alina Su and Dr. Daniel Kohane. Alina is the founder and CEO of NovaXS Biotech, a startup focused on advancing needle-free drug delivery to facilitate the at-home administration of intravenous drugs and to improve the long-term health of patients. Her passion to transform drug delivery into a needle-free, easy-to-use approach has led her to file patents in China and in the United States and to start her own company. This is pretty amazing since she is still a PhD student at Harvard Medical School. In fact, she is a neighbor of Dan Kohane, I understand. Well, Dan Kohane is professor at Harvard Medical School and vice chair for research in the Department of Anesthesiology at Boston's Children's Hospital. He runs a very large laboratory that studies drug delivery. And in fact, drug delivery by biomaterials and nanotechnology. While at first glance, it seems like these are three different things. In fact, they really, really complement each other because when you have a drug that isn't orally available, sometimes you have to use biomaterials to encapsulate it. Sometimes you have to use nanotechnology to encapsulate it, or sometimes you need Alina's approach. So Dan's research also addresses not just drug delivery, but drug delivery in a large range of medical problems, such as pain, diseases of the eye and the ear, and cancer. And he's using a lot of tools to get to his goal. The two of them will discuss their work that transforms medicine in a way that the old needle cannot do. They're proactive and personalized solutions for chronic diseases. So Dr. Kohane will start out setting the stage for various approaches and the evolution of drug delivery. And Alina will then specifically tell us how she started her company, how she is funding it, and how she is developing her needle-free approach to inject drugs. They will both speak for 20 to 25 minutes. After that, we will have questions and answers. And please do type your questions into the chat or the Q&A, and we will then have a discussion and answer your questions. Thank you very much. And please then start your talk.
All right. If you'll allow me to share a screen. Great. Well, I'd like to start off by thanking whoever needs to be thanked for the fact that I'm here today to present to you about drug delivery. And what I'm going to do is basically show you 4 very short vignettes on alternatives to conventional ways of delivering drugs. And just by way of disclosure, I am a founder of a company called Theroptics, which makes drug-eluting contact lenses. And I need to tell you this because I actually have equity in it. And also vignette number 3 was actually the basis for Theroptics. So the first thing I want to talk about is triggerable nerve blockade. And so the background story to this line of research is that about 10 to 15 years ago, a dental filling that I'd had since adolescence was starting to bother me. And I finally convinced my dentist to have a look at it. So he injected me, I suppose, at the superior alveolar nerve with a local anesthetic, which made part of my face numb. And then he took the filling down, and there was this enormous cavity there. And he saw a crack running down the bottom of it. And so he took out his drill and he shaved down the crack, making the cavity even bigger, put in an even bigger filling. And when he was done, he told me in that wonderful euphemistic way that dentists have that I might have little sensitivity later that evening. So later that evening was actually my wedding anniversary, and I was out at a restaurant with my family. And as we were leaving, suddenly I felt like someone had rammed a spear through the roof of my mouth. And what that was the local anesthetic the dentist had injected me with wearing off. And I remember thinking, wouldn't it be nice if when he had injected me, he had injected me with something that I could turn back on? And that led to this line of research. So the idea was to develop a formulation that would be initiated by a single local injection, like an injection at a nerve. So it's not a systemic thing, it's not an opioid, and that it potentially would be quite long lasting. But importantly, the patient, in this case, me, would determine when they get pain relief, that is local anesthesia, how intense that anesthesia is, and how long it lasts. And in the last 10 to 15 years, we worked on a variety of solutions to this problem. I'm just going to share one with you. So here, what we created is liposomes that contain drugs. So a liposome is basically a small particle made out of fatty materials, and it's sort of organized like there are these layers of lipid bilayers arranged like an onion, like in the skin of an onion. And so the drugs that were inside it were tetrodotoxin, which is what kills you if you eat fugu sushi that's not prepared properly, but it's also a very powerful local anesthetic. And it was co-encapsulated with dexamethasone, which we knew increases the efficacy of tetrodotoxin. And so we put these two compounds into this liposome, and this liposome, the lipids in it had a lot of double bonds in it, and that becomes important. Also inside this liposome were photosensitizers, and these are molecules that, when they are irradiated with light of a particular wavelength, produce reactive oxygen species. So the idea was that you'd inject these particles near a nerve, and then you would shine light, near infrared light, onto the site of injection, and that would make the photosensitizers produce reactive oxygen species, and those would peroxidate the double bonds that I told you about, and that would poke tiny holes transiently in the membrane of the liposomes, allowing drug to leave and therefore cause nerve block. And these are data to show that this approach works. And the name in the bottom right is Alina, who did this work. And so here we injected these particles at the sciatic nerve. The sciatic nerve is the nerve that runs down the back of your leg. And so we injected them at time 0 on this graph. And so the x-axis is time after injection, and the y-axis is the degree of anesthesia achieved. 2 seconds means there's no anesthesia, and 12 seconds means there's complete anesthesia or nerve block. So I'd like to draw your attention to the red line. So you can see that after the initial injection, there was approximately 40 hours of nerve block, so about 2 days. And when it wore off, we shone a light onto the site of injection, and you can see nerve block came back. And then after a number of hours, it wore off. And we did this repeatedly with admittedly diminishing returns. Over the next day, we could do it 9 more times, adding approximately 1 day to the total duration of nerve block. And, so we were pretty happy with this. And so this could, for example, replace the prescription of opioids, which is commonly what is done these days in the context of this kind of pain. And we also found that if you replace the photosensitizer with a sonosensitizer, you could actually trigger nerve block by application of ultrasound. The second vignette I'd like to talk to you about is how to get drugs to the back of the eye, meaning the retina and choroid and places like that. And that is generally pretty difficult. And what is commonly done is that people stick a needle into the vitreous humor, which is the gel-like material in the posterior segment of the eye. And so we were wondering if there was an approach where you would not have to do this. I'm actually going to show you 2. And the first one is this. So this idea, those originated with Tal Dvir in my lab was that we would find a way to inject nanoparticles intravenously in such a way that they would go throughout the body, but they would only stop wherever you shine a light. And so we implemented this in the context of ocular delivery as follows. The idea here was to inject nanoparticles intravenously, as you can see in the lower left-hand corner, and the nanoparticles would course throughout the body in an inactivated state. But when they pass through the eye, which is after all built to accept light from the outside, we would shine light in, and that light, when it hits the nanoparticles, would activate them in such a way that they combine the surrounding tissues. And so this is how that works. So the nanoparticles are what's called micelles, which in fact, they're polymeric micelles, and they're made out of the squiggle you can see on the bottom there. And it's composed of PLA, which is hydrophobic, it doesn't like water, and PLA, which is the blue thing, which is hydrophilic, it likes water. And when these things are put into water, they self-assemble into these micelles, which are these nanoparticles, with the PEG on the outside and the PLA all clustered together in the middle to get away from the water. And the PEG was modified with a targeting ligand called a cell-penetrating peptide. And this is the thing that makes the nanoparticles latch onto and penetrate tissues. But in the inactivated state, these CPPs are prevented from doing anything by binding them with this molecule called DEACM. Furthermore, DACM, being very hydrophobic, actually drags the cell-penetrating peptide into the core of the nanoparticle, making it doubly impossible for it to interact with tissue. So the idea is you'd have these things flying around in the blood, but wherever you shine a light, the light would cause the DACM to come off the CPP. And when it does so, the nanoparticle deploys these PEG arms with the CPPs on the end of them, and it binds tissue. So we did this a model of choroidal neovascularization, which is a model that's often used, for example, to mimic macular degeneration and similar diseases. And in the left-hand panel here, you can see where we did laser coagulation of the back of the eye and ruptured Bruch's membrane, which is the innermost layer of the eye's vascular layer. And then 1 week later, we inject, once the injury had progressed with new blood vessels, we injected these animals intravenously with the nanoparticles I just showed you. And what you can see from the right-hand panel is that the combination of using nanoparticles and using this photo-targeting strategy that I described greatly increased the accumulation of those nanoparticles in the tissue, and therefore increased the therapeutic effect. I don't have the time to show you all that, but we showed that if you load it with drugs, it actually can treat the condition. And this work was done by Yanfei Wang. So again, that is an alternative to shoving a needle into someone's eyeball. And the third technology I want to talk to you about, which I co-developed with Joseph Ciolino at the Mass Eye and Ear, is a drug-eluting contact lens. So it is a contact lens that is pretty much like a regular contact lens or a bigger lens that's called a bandage contact lens, except that there is embedded completely in it this donut-shaped ring that is a drug polymer film. So the drug polymer film is not in contact with the patient's eye or eyelids, and also because it's a ring, the patient can see through it. And because this drug polymer film is relatively large, it can contain a very large amount of drug, and also it releases it over a very long period with relatively constant kinetics. And the reason we came up with this originally is because eye drops are very inefficient in terms of how much drug gets in, and also notoriously people tend to forget to take eye drops as prescribed. So this is one example of some of the research we've done with this. We have actually used it to deliver antibiotics, antifungals, steroids such as dexamethasone that you see here, local anesthetics, a wide range of drugs. But here, these data show the delivery of dexamethasone, which is a powerful anti-inflammatory agent. And so we put this lens on the eyes of rabbits, and then at predetermined intervals spanning a week, we left the lens on for a week, but at predetermined intervals, we determined the tissue level of dexamethasone. One thing I want to point out is that the y-axis is actually a log scale. So each major bar is actually a 10-fold increase in delivery. So if you start in the upper left corner in the cornea, you can see, not surprisingly, this thing led to the delivery of an enormous amount of dexamethasone to the cornea. And you can see it's much more than the lens is the dots in black. The boxes in gray are a comparison with dexamethasone drops, such as are commonly prescribed, administered every hour for 8 hours. So you, with this lens, you got a lot of delivery, perhaps 100-fold more than with drops, and it lasted for about a week. What was a pleasant, not altogether surprise, but a pleasant affirmation of a hope was that actually we got a lot of drug into the choroid and the retina, which are all the way in the back of the eye. So what we have here is basically a topical device, something that's put on the front of the eye that that gets drug all the way back to the very back of the eye. And I don't have the time to show you this, but in fact, we showed that it can be used to treat disease models in the rabbit. So this is clearly better than conventional drops. And I would argue that it's better than getting a needle in the eye delivering dexamethasone. It probably as efficacious, but much more pleasant. And finally, I would like to talk to you about treating middle ear infections. This is work done by Rong Yang. And probably everyone in this audience has either had an ear infection as a child, or you are parents of children who have them. It is extremely prevalent in the West. Essentially, all children get them, and about 1/3 of children have 6 or more episodes. And because it is so prevalent, it is actually the #1 cause of prescribing antibiotics to children. And a typical course is 5 to 10 days, usually 10 days, of giving an oral antibiotic 3 times a day to a toddler who's generally really not into it, and it involves a fair amount of wrestling. And that is why there's actually quite poor compliance, and people often abandon treatment once the kid looks better, which is actually not the thing to do. Also, because it's given orally, you are actually filling the entire body with the antibiotic, and which is a little inefficient when you consider that you're trying to just treat 2 tiny areas on either side of your head. And so you get all these systemic effects, that you get rashes and vomiting and diarrhea. And these things can actually be bad enough that care is interrupted, which again is not a good idea. But a final effect of this, of treating such a large number of patients with antibiotics, even though parenthetically, very commonly, the problem is not a bacterial infection at all, but a viral one. The problem is that this can lead to the development of antibiotic resistance, which is actually a major public health problem. So to address all of these issues, we developed the following. So we asked ourselves, wouldn't it be nice if at the time of diagnosis, the pediatrician seeing otitis media on exam, placed onto the eardrum a gel containing a full week of treatment. And so the idea here was to design it so you have a gel that runs fluidly through some device or another onto the eardrum and then quickly gels firmly so it can't be dislodged. And this would contain the antibiotic. The problem is the eardrum, which is actually very similar to skin in structure, is essentially impermeable to most drugs. So inside this formulation, we included chemical permeation enhancers, which are molecules that enable the transport of other molecules across biological barriers. And so, as you can see here, this made it more permeable to drugs, and that allowed local anesthetics also, but antibiotics to penetrate across the tympanic membrane into the middle ear. And what we saw with here non-typable H. flu, which is a bacterium commonly involved in otitis media, but we've also shown it with strep pneumo, which is another bacterium very common in otitis. We got in chinchillas, which is a standard model of otitis media, we got 100% cure rate with complete eradication of bacteria in the middle ear. And these data basically just show you what I just said. So you'll recall that earlier on I had said that there's all this concern about the systemic delivery of the antibiotic. And if you apply it directly to the ear, maybe there will be no systemic delivery. So what we did is we measured blood levels in these chinchillas. And what we found is that over the course of the week, there was no detectable antibiotic, that's ciprofloxacin, in plasma at any time point. So basically, what we have here is a formulation which would replace oral antibiotics and all their problems. In closing, I just want to thank my lab. This is at this point a pretty old picture, but a lot of the people whose research I showed here were actually involved back then. And thank you for your attention. And if there are questions, I will answer them after Dr. Su's presentation.
Thank you so much, Ben. And I would like to introduce Dr. Su, or not yet Dr. Su, but working on it. Please go ahead, Alina.
Awesome. Let me share my screen. Am I sharing my screen?
You were sharing. There you go. It's back.
There. Awesome. Awesome. Great to meet everyone. And very great presentation from Dr. Kohane. Very excited to be here and great to talk to you about what we are doing. Very exciting stuff, which is needle-free injection technology. Like, everyone hates needle when you are need to inject yourself for 10,000 times over a year. So we started this company because of background of my family. They were having diabetes, which require injection for 1,000 times into their belly in one single year. So what we are able to see is that all of the belly is with hard knots because the aggregation for 1,000 times of injection, and that is getting their skin harder and harder. So that will be preventing themselves to actually able to absorb any of the insulin anymore. So the insulin dosage have been increased up to 3 times up to this day for 20 years of injections. And that is what shouldn't be done until this day. So that's the reason that we have started NovaXS Biotech for patients who need frequent injections at home with chronic disease. So what we're trying to solve is not only poor drug delivery instrument that will solve the pain and fear of the needles, but also eliminating any of these negative side effects that will be caused by needle itself, which with 56% of the high administration errors and also low medication adherence, as we were saying, nobody wants to inject themselves for 1,000 times over 1 year. That's why over 40% of chronic patients are not adherent to the treatment. So to resolve that, we just released RPN devices. So from 2021, Medicare Plan B have released remote therapeutic monitoring and also remote patient monitoring code. But that adaptation rate is only for 10%. So in that case, medication adherence data has not been collected. And at the same time, the biosimilars and generic drug is facing a bigger problem that when patients are not injecting themselves, they need to do that in their home. And there needs to be a better solution for IV therapy, where patients need to inject themselves at home rather than getting into hospital, sitting there for 2 hours with nurse shortage, but also doctor shortage. So why is this important? Like, how severe is the medication non-adherence? 6 in 10 Americans is having at least one chronic disease, but 40% of the patients are not compliant. And that is causing the financial loss of $290 billion avoidable cost for US government due to medication poor adherence and chronic complications. And that is also resulting $637 billion revenue for the global pharma companies that forfeits each year. And that also for patient themselves is increasing $2,000 as insurance company, plus 3 visits for non-adherence patient annually. So in that case, what we have built is a very interesting system called needle-free injection jet delivery system. And that is allowing us to deliver medication within 0.3 seconds by generating a very thin stream of high pressure jet injection directly into subcutaneous or intramuscular level. So as you are seeing here is our prototype. So what it's doing right now is it's able to get in the medication from any of the bio with the accuracy of 0.1 milliliter, all automatically, and able to use repetitively for 10,000 times. So what patients need to do is to press the button, and that will be delivered into their body without the needle, purely by the pressure. So at the same time for needle-free injections, when they're pressing, we'll be sending the signal directly to our patient management system and also the app. So Talasys, it's a integrated medication adherence system that not only delivers medicine, but also tracking the medication adherence and also dosage volume data. So look at here, it's mainly targeting 76% of the patient's treatment progress is completed, and that what is their adherence rate to the injectable data. Also, it will be synced with other biomarkers to evaluate health risk in the second page. So in that case, for diabetes, the most common side effect that we are seeing is heart rate related disease. So in that case, if we are seeing an insulin level increase, but blood sugar level is not decreasing, that potentially means there's already side effects going on. So at that point of the stage, insurance company or doctor could be using preventative method to start to measuring how patients are doing and start to do measurement upfront before it's actually happening. And third one is a valuable real-time data for patients and providers and able to be communicating with each other to solve the preventative care, but also early interventions. And here is some functionalities that we are able to be delivered with needle-free injectors with volume up to 1.0 milliliters in 0.3 seconds and viscosities up to 33 CPs. So that means very viscous medications, including hormone-based level, oil-based level, like progesterone oil in vitro fertilization, that could be also able to deliver for needle-free injections. And in terms of tissue layer, we are able to deliver into intradermal, subcutaneous, and intramuscular. And that means we have the capability to inject a wide range of medications with automatic medication loading system and dosage verification. But this is not only the very exciting thing. The most exciting thing is that we could be repetitively using this system for 10,000 times for automation for medication loading, but also injecting. So that means not only we are able to have a better delivery rate for patient experience, but also at the same time, having much more sustainable way by filling the medication into one single disposable vial like this. So in that case, how are we able to benefit the entire remote therapeutic? For practice beneficiary, it will be treating from the base assumption how patients are adhering to the medication into very informed decision and early intervention based on real-time data. But for pharma, they will be having a much more better prescription rate and reducing their cost of goods sold by 37% because it's sustainable and repetitively usable compared to what they're using right now, which is the auto-injector. And for patients themselves, we're able to eliminate the pain and side effect that caused by needle and lack of information into a much more friendly system that will support users' journey. And for insurance healthcare system, not only we're able to track the adherence and assume 80% of the intake, but we are able to collect much more accurate qualified medication system with IoT sensor to collect the adherence data. So how exactly are we able to directly beneficial for pharmaceutical partners? We can directly see with our cost of goods sales compared to auto-injector, which is using different very famous drugs like Humira, Triggerable, or Trulicity from Eli Lilly. So the total annual savings, because we are not disposing the entire medication delivery system, but only disposing this vial that will be pre-filled or automatically filled in patient's home, that will be having a total annual savings of 34 million, 150 million, 61 million, only solely by changing the delivery system from needle into needle-free injections. And that market opportunity is huge, as we all know, for target therapeutic areas, including diabetes, severe obesity, GLP-1 medicine, severe asthma, rheumatoid arthritis, IBD diseases, migraine, IVF. What we are targeting is plus data monetization market potential of in total of 103.5 billion of the entire huge market, with 82.5 million of US population who is having chronic disease, who need frequent injections, daily or weekly. So let's take a look at some very exciting things, which is our preliminary data that will be very adherence and also especially for diffusion rather than injections. So when we're taking a look at the upper right system, what we are able to see is that the delivery rate compared to traditional needle versus needle-free injections is much more able to using diffusion with pure pressure jet rather than injections. So that also means the adherence level will be increased, but also at the same time, the improved absorption rate has been proved according to our clinical study. And the mechanism of that is that by using pressure to press in the medication into human body, we will have a larger contact area between the medication and human body. And that time, we will have a better absorption rate. So let's take a look at the data in boosted efficacy with needle-free injector, which is a 16-week multi-center randomized clinical trial with HbA1c insulin injections for 412 patients enrolled in the clinical trial. For needle-free injections, what we are able to see is that the decline of the HbA1c is much more significant compared to syringe because the absorption of the insulin is much more better compared to traditional syringe. And that is sitting the exact same in different functionality and also different parts of the body, including thigh, abdomen, and also upper arm. So the infusion rate of the curve is very similar in different part for needle-free injector. And the injection depth is able to be directly controlled by what is the mechanism of the pressure that will be delivered. And what we're also able to see is the fewer skin irritation and negative side effects that will be having less painful compared to needle injections. So take a look at figure A, that is proving red and swollen and also bruise. Compared to traditional needle-free injection is having a better patient experience. And also with the increase of the number of injections, that is becoming more and more significant, what we are able to see with a 14 days randomized clinical trial with 62 different patients compared to traditional needle and needle-free injections. So in C, what we have tested out is uncomfortable feeling, operation convenience, the fear of injection, and also the accessibility. So in this case, specifically for chronic injection patients, the fear of injections has been declined very severely, and this will be a much more better efficacy to increase their adherence level to the medication injections. And when we are taking a look at these pictures, what they are showing is the pain score and pain percentage compared to traditional needle and needle-free injections technology. So it's much more less than traditional needle by 2 times. And the pain percentage of the needle-free injections is usually from 0 to 3. And for general needles, it will be much more higher, which is usually 78% of the patient declared it will be a 6 points for a traditional needle, which is 3 times larger than needle-free injections. So what are we up to right now specifically? And what will be the next wave of needle-free injection coming to part? So for us, right now, the cost of goods sold for us to producing is about $450, reusable for 10,000 times. But with massive production, they'll be able to decline into $150, which average cost of per injections will be $0.7, which is less than $1 per injection. And in that case, with example of weekly injections for 2 million of patients, that will be saving 21.8 million of the dollars. So that's not only a better adherence rate, better experience, but also cost of savings, not only for patients themselves, but also for practitioners and also for pharma companies and insurance providers. So that is mainly what we are targeting. And in terms of competitive landscape, there is several needle-free injectors on the market. For PharmaJet, what they are targeting is a vaccine injection in developing countries such as Indonesia and also Africa. But in terms of their cost of goods sold, they are very expensive because they are targeting in hospital usage only provided by providers. And for CrossJet, they are pre-filled only single usage needle-free injector, because what we are able to see is that needle-free injector is having a much more faster, fast-acting injection system. So that means medication will have a better absorption rate compared to traditional needle. So they're using in ER specifically for rapid release medications. And compared to auto-injector and pre-filled syringe, what we are able to provide is much more usability with sustainability, process automation, repetitively injecting, and also with the same time adherence tracking with pre-filled or vial storage of packaging of medication. So in this case, what we are hoping is that everyone is able to get one device at their home, and we will be shipping the vial as a subscription model to provide to them a much more better delivery way of medications. So here's our outstanding team, including very experts, former president of 10 hospitals, Sean, and IRB clinical study specialist, Haley, is also our VP, and 30 years of experience of sales with his Jim. And we are currently backed by Baxter, who incubated us, and also the largest pharmaceutical company with medical device manufacturer. They are the actually very first needle productions. So they're the top 1 needle productions for the entire country. And they are our first check. So it's very proud that they're thinking of shifting into better delivery way, replacing the needle. But at the same time, we are currently in Techstars working with Cedars-Sinai and also United Healthcare with providers, but also leading investors like Berkeley Skydeck and also Edward Elms Hospital. So in that case, I will end our conversation here, and I hope we will have a better delivery way so the patients like my family will not need to inject themselves for 10,000 times with needles and suffering from all of the pain and medication aggregation with all of the injection side effects. Thank you.
Thank you so much, Alina. This was very interesting, and we heard now of several delivery models. And I have some questions here, so let's get started. The first question is asking whether for diabetes, the implantable pump with continuous glucose monitoring isn't competitive.
That's a very good question, Ben. So in terms of what we are able to see in implantable pump for diabetes, it's usually having a side effect that will be used within 3 to 4 years, and people need to shift back into needle injections because it's not able to do a frequent delivery. So that is a usual side effect what we are seeing with implantable pump. And another area why it's not able to do a go to market very quickly is that the cost of goods sold is too high. So for markets that are in Europe, it's covered by the insurance company. But for markets such as in Asia or US or Canada, it's not 100% covered by insurance. So that's why the cost is very high for competition in terms of implantable glucose pump.
Thank you. And the next one is also for needle-free injection. How close are you for commercialization? You have treated over 500 patients, and you have measured biomarkers, you have measured absorption. So how close are you?
Yeah, so we're about 1 year to commercialization. So we count as class 2 medical device with 510K with predicate. So that means as long as we're proving safety and consistency in US, that will be enough for us to get the clearance, which usually takes 6 to 9 months. But to be very safely, we are targeting a 12 month go to market.
And that's in the United States?
That's correct.
Wow, okay. And then somebody remembers history. He is asking whether the US military used a similar technology for vaccine administration 50 years ago.
That is correct. And that's a very interesting story behind it, because 50 years ago, there was huge contamination for blood transmission. And that is the reason why needle-free injection didn't get adopted into the medication administration system. But the reason why there was contamination is because there was a backflash of the blood between different people. So in that case, if the needle-free injections has been administered to me or to anybody else right after, without changing the nozzle at the front, that will be directly causing the contamination because of the black flesh of the blood. So what we are targeting is that one single-use drug administration, which is the vial that I just showed to everyone, that will be a single-use doses administration, and that will be disposable because that will be pre-filled with the medication or directly draw the medication into that vial. And that is single-use to eliminate any of the contamination or transfections.
So let me have a follow-up question. So that means that one pump cannot just be used on different people, but also for different drugs.
That's correct.
You said there is no contamination because whatever you have on front is disposable.
That's true. So in that case, we're working with different drugs like Humira, GLP-1 medicine, PD-L1 medicine. So a lot of medications is actually shifting from different vial, for example, a vial that is in IV injection into subcutaneous injection. So that also means we could have a very wide target list for different medication, but also different viscosity of the medicine for delivery.
Now, you are injecting pretty large molecules just by blowing air. Do you see a gradient between the size of the molecule and the absorption? Like small molecules are a lot easier to administer, and the larger the molecule, the more force you need.
Mm-hmm. So what we are seeing right now for testing is that the limitation factor is usually by the volume. So what we are able to be delivered up to the date right now is it doesn't depend on the molecular weight or molecule size. It's by the total of the volume it needs to be delivered. For example, right now, the limitation is 1 milliliter for a single dose administration. But if we need to change it into 2 milliliter in total of the dose, that will require much more bigger pressure, but at the same time, a larger vial that will require the safety and also consistency delivery.
Okay, I think we have two questions that are similar. Okay, the one is for Humira. When do you think you're going to have a Humira delivery system?
We are trying to talking to the developer of Humira, who is AbbVie, and that is the current.
Teva who came out with, isn't it
Yes.
Teva that has the generic?
Yes, Teva also have the generic for Humira. So that is what we are targeting right now. We are actively doing business development with pharmaceutical company, trying to tell them what is available, what are we able to do, and trying to collaborate with their medication. So if anyone has connections to pharma, we will be loved to talking.
Okay, I think there is another question that kind of addressed is how much cleaning is needed. Because you said that most of it is disposable, but you could still have some contamination on the rest, right?
Yeah, so no cleaning is needed because it's directly sealed in a one single dose vial. So in that case, there is no any of the cleaning needed.
Okay, I have a few questions now for Dan. And one is actually mine. You said something like a lot of otitis is viral. Then what do you do about that? You just give the antibiotic, or do you want to have a test first to see what it is or it's not?
Depends where you live. In Netherlands, they do nothing. And here, the consensus from the American Academy of Pediatrics is that everyone gets antibiotics.
Independent of what it is.
Well, the problem is that there's no way to know what it is.
Why can't you take a ear sample?
Yeah, so the idea of shoving a needle through the eardrum into
You have to put it through. Got it.
yes, yeah, most mothers would not go for that, and the children even less.
I understand. I thought maybe you could take it from the outside. Okay.
That would be very cool, but we're not there yet.
Okay. And then somebody is asking, can nanoparticles be used to deliver chemotherapy to areas near the optic nerve?
Yes. In fact, my group is working on that. Yes. In principle.
But what you had in your lens are not nanoparticles, or are they?
No. No, but so for example, in the technology I showed where you inject stuff intravenously, if you just shine if you shine the light on the optic nerve, they'll go there.
Okay. And then the same person is asking about delivering steroids.
Yes. Sorry, what is the question?
The same, nanoparticles to deliver steroids to the optic nerve.
Yeah, so I mean, the thing is, if you can get the nanoparticles there, nanoparticles are sort of like trucks. You can load them with anything. That's a bit of an exaggeration, but it's also sort of true. And so absolutely, you can load nanoparticles with steroids, and I've done that. Many people have done that. And then you just inject them and use targeting techniques to make them go where you want them to go.
And when do you think that your nanoparticle delivery system, which, as you said, it's kind of delivering whatever you want to deliver. So you have one for teeth, you have one for eyes, you have one for wherever you want to stick them. When do you think you're going to have nanoparticles on the market for a system?
I don't know. Honestly, the problem is that I have 3 jobs and I have a bunch of technologies that I'm trying to commercialize, and I'm one person, so it's really a bandwidth issue. But we have some nanoparticles that are probably fairly ready to go. Not on the market, but commercialization.
Yeah, so along the same lines as, the people are injecting an anti-VEGF compound, I forgot the name, into the eye for macular degeneration.
Yes.
And that could be delivered with, I don't know, whether nanoparticles or your lens, but if you had something for that, because the once a week, every 2 weeks injection is really not what people like.
Yeah, so it's actually really with that in mind that we developed that nanoparticle targeting to the back of the eye system. It would be great for people. I mean, it's funny, you talk to some ophthalmologists and they say, oh, it doesn't really hurt if you shove a needle in the eye. But I've spoken to some patients who tell you it is amongst the most painful things you can endure. And yes, so very much ultimately that is the hope. With the lens, we don't know but we are somewhat skeptical about the ability to elute a large molecule out of the lens and then all the way to the back of the eye through all the barriers. I mean, the thing about the small molecules like dexamethasone is they actually have properties that are relatively favorable for going places.
Yeah, that brings me back to Alina. You showed, you did not show how your injection system, what you find systemically. Did you measure blood levels of whatever you're injecting?
Yeah, so in terms of insulin, the direct biomarker should be the blood sugar level, the adjustment, the declination of that. So that is what we are targeting for insulin. And in terms of the vaccine measurement, that is usually what we are using antibody rate of the generation to measure what is the efficacy of delivery.
But doesn't the FDA want systemic exposure?
For different medication?
Or anything you're giving people.
So what we are doing is usually compared to traditional needle. So that is the A/B testing in terms of what is the absorption rate and what is the efficacy rate of delivery. So that is usually a A/B test between needle-free injection and traditional needle injections.
Sorry, what is A/B test?
A/B test is a same group of people that will go through same exact dosage of the amount of the same medication, but purely with different delivery methods, such as needle and needle-free injections.
Sorry, I missed something. I'm sorry, I'm condensed. And then how do you look at systemic exposure in a crossover study?
So systemic exposure is mainly directly measured by biomarker that has been clinically validated.
The FDA doesn't want you to measure actual exposure? I'm asking, I don't know.
So for a class 2 medical device, I don't think they are targeting that specific systematic exposure. So it's just a stability and consistency test compared to needle.
Okay. Okay. Yeah, I guess you're in a better field than I am. In our field, we need to show everything. Okay. Yes. Do you want to comment maybe a little bit about how you see the future? So let me start with you. I know you just said that you don't have time for everything, but you have some good delivery systems. So how do you see the future of delivery going? Because, as you know, in some ways, you and Alina both say that sticking needles into people is not that nice a thing to do. And you both came up with alternate ways. So then how do you see future drug development going?
So I'm mindful of a paper that Bob Langer wrote with someone whose name escapes me. I feel terrible about that, about the importance of convergence. And actually, Alina's thing is not a bad example of that, where disparate areas of technology sort of are coming together to have a certain effect. You know, other examples would be, you know, a lot of the stuff that I showed is what's called smart materials, which impart functionalities. On the other hand, the market and doctors in general don't like complicated things.
Right.
And so, you know, in some ways, it's much easier to move forward with a Me Too type compound than it is to go with, like, you know, my triggered local anesthetics.
Right, right.
Yeah.
We have 2 more questions, and while Alina, have to maybe ask you something. Your system does not work in the eye. You're going to blow out the eyeball. Is that correct?
Too bad.
Okay, so it's for Dan. Is there currently an FDA-approved alternative to needles in the eye for the treatment of macular degeneration?
I know a lot of ones that are under development. I'm not aware. I don't know if there are some commercially available. And yeah, I'm not an ophthalmologist, so I don't know.
I kind of don't think so. I haven't.
I don't think so, but I don't want to say for sure. Because as I said, I'm peripherally involved in a number that are under development. So.
And then the very last one is delivery to areas of the brain and interior areas of the brain. This person is very picky. It's very hard to get to interior areas of the brain. Dan, you have any idea?
Yes, so there's a huge amount of research on it. And in truth, it's a little hard sometimes to separate hype from what is real and translatable. Also, very often people show they can get drug there, but it's not clear that it gets there in sufficient quantity to have efficacy. That being said, there are a lot of things that do work. And actually some that are commercialized, a lot of them actually develop on getting into the brain with a sharp object. So for example, there's the Gliadel wafer, which was invented by Henry Brehm and Bob Langer some long time ago, which are these wafers that release chemotherapy, and you put them inside the place where you scooped out a glioblastoma. There's a huge amount of stuff on nanoparticles or drug conjugates that hijack receptors, transporters to get into the brain. Also work where, and this is sort of analogous to some of the stuff I showed, where they use focused ultrasound to temporarily, they say, disrupt the blood-brain barrier so that drugs and particles get through. So there's a lot of work on it. And there's all kinds of things where there's enhanced diffusion devices. Nothing that I'm aware of that is there.
Well, maybe there is. It's the very, very, very old-fashioned hydrophobic, systemically given oral drug. Valium gets beautifully into the brain. Okay?
Yes. No, absolutely. The problem is that it also gets out of the brain.
It's all over, right?
Yeah.
It's all over. I agree.
No, but also it leaves, meaning or it gets metabolized and destroyed. So getting things that last in the brain and that are in high concentration in the brain, not elsewhere, that is the trick.
Right. Okay. Well, thank you very much. And I think we're at the end, and it was really interesting to see where we are going. I don't need a daily injection yet, but trust me, Alina, if I have one, I'm gonna come to you, okay? Thank you.
Will do.
Bye-bye.
Bye. Thank you, Maria.
Sure. Bye-bye.
Bye-bye.